Hi, now we have a topic for biomolecular principles for biomedical engineering. So today we want to study molecular principles in biomedical engineering. So we understand the basic structures of proteins, which are polymers of amino acids, and how the diversity of this amino acid structure influences the protein's 3D structure, the dimension, and its function. And the goal for this is to lead you to introduce the nucleic acid as polymers of polypeptides and how these structure is different in DNA and RNA polymers. And this lead to the importance of deoxyribonucleic acid or DNA in storing our genetic information in our cells. And we will briefly review genetics 101, and that leads to the central dogma of molecular biology and the concepts of biological transcription and translation. So this will lead us to think about the importance of gene cloning and how recombinant DNA technology has revolutionized biology and medicine. So first, let's talk about why DNA is important in human health. So we have heard a lot about DNA. Probably a lot of you already know what DNA is. And in fact, thinking about human health issue, many diseases result from failures at the DNA molecular level. So for example, there could be a defect in the gene, which is in DNA, that gene itself may have a defect which causes genetic disease. However, I would say these are relatively rare. And on the other hand, there could be defect in the regulatory regions of the genes, which produces eventually functional protein, and that function could be problematic. For example, regulatory gene regions can affect the gene which may be related to proliferation, cell proliferation, and suppression of tumor formation, which can cause cancer. So this will be very important because cancer is almost top cause of death in the developed countries. And examples of a single gene defect. What if a single gene can have a defect which can cause a problem? So for example, cystic fibrosis is a single gene defect disease, which currently we don't have any treatment. However, this is a severe that this causes a problem in lung epithelium, and patients can suffer until, so the patients will not live long enough. So that's a problem. Currently, we are developing treatment for this genetic defect disease. And another example is muscular dystrophy. This is about genetic disease causing the muscle weakness. Eventually, it's problematic. And another example is Huntington's disease, which is also caused by a genetic defect. The problem of this genetic defect is the cells affected, all of these cells have a defect, so we cannot produce a functional protein properly, so that it affects the patient. And it's not very easy problem to solve. And another way is how to cure, fix this genetic defect, which requires a gene therapy, which has been progressed a lot recently. And also there's a lot of engineering issues. For example, let's say we have a virus containing to fix these genes throughout our lung epithelium. How do we deliver it? And how can these genes fix to deliver to each of the cells inside the lung in case of cystic fibrosis? So this has been a challenge, and our engineering and scientific approaches will be very important. Let's start talking about building blocks of proteins first. So what are proteins? protein is a macromolecule of consisting one or more chains of long chains of amino acid residues. So proteins are the most abundant molecules, I would say, in composing our body. So proteins are made up of polypeptide chains. So this polypeptide is a polymers of amino acid. Let's see how amino acid structure look like. So amino acid has a first central carbon atom, and carbon has this 4 bonding unit, and one side is a hydrogen. This side is a NH3+, which is amine part, and the other COO minus, this is carboxylic part. And what's determining the name or the structure of this amino acid is this residue group, this R group. So at the unionized form of amino acid looks like this. So CH and NH2 and COOH, and there's a residue. So this unit of amino acid can polymerize to connect to one by one, and that we call as a polymers of amino acids as a polypeptide chain. So this polypeptide should have this amine and carboxylic acid and side group. As you can see, the other, this hydrogen and amine and carboxylic acids are common. So what determines its unique structure, unique function is this side group R. So that R1, R2, R3, these are amino acids, different kinds, and we can polymerize this and we can name it R3, R2, R1, R3. So this R is the determining factor characteristic, and that we call as a polypeptide. So polypeptides are polymers of amino acids. There are about 20 kinds of standard natural amino acids with varying side groups. The properties of these side chains or residues determine eventually the three-dimensional structure of the polypeptide. Proteins are made up of one or more polypeptide chains. While its sequence is kind of linear, but in reality, inside in our body, it actually has a three-dimensional structure. So let's discuss a little more about these amino acid groups. So the property of this R group side chain determines the property of amino acid. So we can regroup them into non-polar and polar and acidic and basic. So non-polar, the characteristic is hydrophobic. Hydro means water, phobic means dislike. So I put one water molecule here, which is H2O. This water molecule, in fact, from its electronic aspect, hydrogen has a partial positive. Partial, we put this Greek letter delta, which doesn't mean it's actual charge, but it's more of a partial polarity. So it's more like partially positive, while the oxygen molecule atom side is partially negative. So this we call as a polar. And in physical point, we can draw something like a polar dipole moment we can assign. So this water already has a polarity. So water likes to interact with a polar residue or polar molecule. So that's why polar molecules will be liked by water. So while it's non-charged, so there's a number of amino acid groups. And one good example is here. This is a threonine amino acid like CH, CH3. You see this OH. So this O oxygen has a polarity, so which would like to interact with water. While the other non-polar molecule, such as leucine, an example, this is all carbon and hydrogen, and these are not polar, so water would not like to interact with this. So this is hydrophobic. So this part of residue inside the water, they may hide inside the structure rather than coming out to directly interact with the water molecule. The other aspect is now the actual charged molecule. So for example, aspartate, one example of amino acid has CH, COO minus. This is actually negatively charged. So aspartate or glutamate, and these are negatively charged, and this will be an acidic group. On the other hand, like this lysine molecule, you see this positively charged NH3+, and this will be a basic group. So many proteins which are composed of amino acid chains or polypeptide are serving as an enzyme catalyzing chemical reactions. Some other proteins are supporting in and outside of the cell. So this is structural support. So what else proteins can do in our body? Can you think of? Yes, antibodies are all proteins. So proteins are the most important functional unit of our body. And this is a kind of nano structure, nano machines in our body. So to give you an idea more detail, I put here amino acid more details. So detailed group so that you know what amino acids are. So amino acids with electrically charged side chain. An example is positively charged. So these are the side chains. And this is an arginine. And we write it ARG, or biochemists, even to save the space and time, they put as a R. So each amino acid has a full name and 3-letter short name and even 1-letter name. So another example of negative, for example, aspartic acid, we write it ASP or in 1 letter in D. Honestly, I'm not a biochemist. I don't remember this, but there, biochemists use this single letter to represent one single amino acid. Another example is polar uncharged side chains in serine, threonine, asparagine, and glutamine. And you can see this OH, and these provides a polarity, negatively charged. And special cases are here cysteine, glycine, proline, and hydrophobic side chains, alanine, ALA, A, valine, isoleucine, leucine, methionine, phenylalanine, tyrosine, tryptophan. And these are ones you study into biochemistry or more deeper, and you will face it a lot of time. So this is in a nutshell, a single amino acid will have a structure like central carbon, and here an hydrogen molecule, and one side is amine NH2, the other carboxylic COOH. And what determines its name is actually the residue over here, side chain. And this one amino acid and the other amino acid, you can see this carboxylic side COOH, this OH and H from this amine, and this H2O will come out as a water molecule, and the rest can be connected. The CO, you can see CO, and this NH, CONH, and they become a polypeptide, this peptide bonding. So this is peptide bonding, and it keeps making a change of amino acid.